Today we will discuss about the in vivo study of CAR-NK-92 or 5.28Z that is used to target the HER2 antigen that are usually found on the breast cancers. So what it is, they take the immunocompetent mice and inject them with this tumor, this GL261 or this ErB2 in the right brain. Then after the 7 days they started the treatment with intratumoral injection of these NK cells. For control they used wild type NK cells, and for the studies they used CAR-NK-92 or which they call them as 5.28C, and they inject them weekly for 3 times, and after that they are able to see the tumors is removed or tumors is eliminated. And thereafter they also did the rechallenged on the left brain, and in the first rechallenge they are able to see that 5 out of 8 mice shows tumor rejection, and whereas when they use the wild type NK-92 they none of the mice shows tumor rejection. And in the second tumor case all the 5 mice shows tumor rejection, whereas the naive mice which are not undergone any treatment does not show any rejection. And thereafter what they did is they took this mice that shows tumor rejections and then use this anti-CD4 and anti-CD8 to prevent the T cells effect, that is CD4 and CD8 effect on the tumors. And after that when they inject the tumors they are able to see that in most of the cases the tumors are able to grow into the mice even though they are previously treated with CAR-NK-92 or 5.28C. So from that they come to the point that this CAR-NK-92 cells will cause apoptotic of these tumor cells, and this apoptotic of the tumor cells will then go to the will help this dendritic cells to transfer this antigen information to the T cells that will help in the adaptive immunity developed against the tumors by these T cells. And in order to validate that, they check the antibodies in the mice which are treated with NK-92/5.28C, and they are able to see the presence of not only this ErB2, which is because of which might be because of this NK-92/5.28C, but they also see the antibodies against GL261, indicating the antibodies generated against the tumors, not just the antibodies that are present in this CAR-NK-92/5.28C. Later on, what they did is they depleted this CD4 and CD8 antibodies. We can see that before depletion there is a presence of CD4 plus and CD8 plus antibodies, but after depletion the CD4 plus and CD8 plus antibodies goes down. And thereafter, after this mice is injected in this case, and they are able to see that in the mice where there is no T cell depletions, the MRI looks clean, whereas in the case where CD4 plus and CD8 plus T cells are depleted, the tumors is again starts to regrow, and that is confirmed with this MRI. This MRI has more white extra edges and compared to the control case. So this in altogether indicates the interplay between CAR-NK-92 and T cells that we have discussed in the previous video, where we have seen that this CAR-NK-92 not only helps in this innate immune response, but it will also help in the adaptive immune response that is generated against the tumor cells, and it can last longer because these memory T cells will last longer into the patient. And this is one of the reasons why during the first rechallenge and even after the second rechallenge the researchers are able to see tumor rejections in the mice which are treated with CAR-NK-92/5.28C. So I hope you understood, and these things they further validate that this adaptive immune response is because of the T cells. By depleting the T cells, and then they are able they rechallenge the tumors, and tumors are able to grow. So this indicates the interplay between CAR-NK-92 towards this adaptive immune response and innate immune response. I hope you understood how they validate their hypothesis that they have made in the previous videos using this in vivo experiment. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.